morning, dear students. Welcome back to our official YouTube channel of our college, Saran BGNR Arts and Science College. And this is Dr. B. Satish Kumar, Assistant Professor in Biochemistry. And this is the last video lesson of third unit in molecular biology, which is the paper 7, core paper of final year biochemistry students. In last few video lessons you have learned about the basic processes of regulation of gene expression. Okay, so we finished studying what is induction and what is repression. So for induction, that induction is explained by taking an example, well-known example, that is lactose operon. And for repression, for the understanding of repression, we have taken an example of tryptophan operon. Okay, so after that we have also studied what is known as catabolite repression. In the just previous video, you have learned about the catabolite repression. Okay, so in the catabolite repression, that glucose is going to repress, the presence of glucose is going to repress the activity of lactose operon. Okay, so that repression is mediated by cyclic AMP levels. So if the medium is having both glucose and lactose, both the things are there, if both the things are there, glucose and as well as lactose in the medium, then what happens? The glucose levels will cause the decrease in cyclic AMP levels. Okay, so because the cyclic AMP levels are less, so then what happens? The activator site, that is cap site in lactose operon, will be unoccupied by the activator protein, that is CRP protein. Okay, so and lactose is present, because of that operator site is unoccupied, because that repressor is gets dissociated from the operator site once it binds the lactose. Okay, so in this case the cap site is unoccupied, therefore a very low level of expression takes place. So that is almost like switching off. Okay, that operon is switched off in the presence of glucose, even though the lactose is present along with the glucose. So glucose is now repressing the lactose operon. Okay, it is dominating on the lactose. So as long as glucose is there, the lactose operon will remain in switched off form. So once the glucose depletes, then only that cell start utilizing lactose as the energy source. So this is what catabolite repression that we have discussed in the previous video class. And now in this class we are going to finish the remaining topics of this unit, that is nothing but attenuation, tryptophan attenuation. Okay, transcription attenuation. So we are going to take the example of tryptophan operon once again to understand the transcription attenuation process. Okay, so before that I will explain once again about the regulatory process involved in tryptophan operon regulation. Okay, let us quickly cover the functioning of tryptophan operon. So as everybody know about the tryptophan is one of the amino acid essential for the synthesis of polypeptide chains and proteins. So therefore tryptophan must be synthesized. So not only tryptophan but all the 20 different amino acids must be synthesized continuously, because they will be continuously utilized by the cells. So because the translation process is not an occasional process, it takes place throughout the life cycle of any organism. Okay, not only the prokaryotes but eukaryotes, higher level organism, anything it may be. So almost all living creatures will continuously express one or more genes. Okay, so that is why for the production of proteins the amino acids are very essential. Okay, so that is why that E. coli cell continuously synthesize all the 20 different amino acids. So one among the 20 different amino acids is tryptophan, so it should synthesize the tryptophan continuously. Okay, so that means that genes, the structural genes which are required for the biosynthetic pathway of tryptophan must be always in switched on form. Okay, so if the levels of tryptophan goes beyond a particular limit, that means if they are abundant, if the supply of the tryptophan or the production of tryptophan levels is abundant, then only that tryptophan operon is repressed. Okay, so it always try to meet the demand of tryptophan, consumption of tryptophan. So if the consumption is less and production is more, then what happens? The quantity of the levels of tryptophan goes up. So now temporarily it should be slowed down. It should be slowed down to some extent. Okay. So once the levels goes down again, then it should increase the transcription process or expression process once again. So that kind of regulation happens in case of tryptophan operon. Okay, so the tryptophan operon is having 5 structural genes. Okay, let us look at the diagram showed in this slide. We have 5 different structural genes A, B, C, D, E. Okay, in reverse direction. So these 5 different genes, when they are transcribed, produces a polycistronic mRNA. From the polycistronic mRNA, 5 polypeptide chains will be synthesized, and first 2 polypeptide chains are the components of a large enzyme, that is anthranilate synthase. Okay, so first gene produces component 1 of the anthranilate synthase, and second gene produces component 2 of anthranilate synthase. So 2 component 1s and 2 number of component 2s interact, assemble to form a multi-subunit protein. Okay, that is anthranilate synthase. And from the third, that is tryptophan C gene, it will get the indole 3 glycerol phosphate synthase enzyme. And from the tryptophan B and A, from the B it will get tryptophan synthase beta subunit, and from the tryptophan A it will get the tryptophan synthase alpha subunit. So 2 alpha 2 beta subunits associate to form one more multimeric enzyme, that is tryptophan synthase. So we have 3 different enzymes which participate in the synthesis of tryptophan from chorismate. Okay, from chorismate. If the tryptophan if the levels of tryptophan is more, so then what happens? The tryptophan regulatory gene that produces repressor which is inactive. Okay, which is inactive. The inactive repressor it produces. Once the tryptophan levels are more, that high level of tryptophan, that level comes, that tryptophan binds to the repressor and make it active. So the active repressor binds to the operator site and stop the transcription process of structural genes. So therefore it switch off. So if the tryptophan is more, so then the tryptophan acts as co-repressor. Now the co-repressor binds to the repressor and forms a complex, that is active complex. That active complex binds to the operator site and stop the transcription process. If the levels are less, so then what happens? The tryptophan repressor synthesized is already in inactive state. Okay, so that is unable to bind the operator site. So levels of tryptophan is less, so the tryptophan won't bind this tryptophan repressor. So this repressor cannot bind to operator site. So therefore the transcription of structural genes takes place, and ultimately that leads to the production of enzymes necessary for the production of tryptophan amino acid from the precursor molecule, that is chorismate. Okay, so that is what the regulation process. Okay, so and here the fourth topic and last topic of this unit is transcription attenuation. Attenuation means here stopping. It is also stopping or inactivating. Okay, premature termination of transcription, in other words. Okay, transcription must be attenuated. Okay, not at the beginning, but the transcription process starts, but before it is completed, it is prematurely stopped. So that is what it is known as the attenuation. So premature termination of transcription takes place. Okay, so that is what transcription attenuation. So here the tryptophan operon is regulated by two mechanisms. So first mechanism we have just mentioned, and that is by tryptophan operon. So that repressor co-repressor complex that will regulate the tryptophan operon. It's just a normal repression process. Okay, so that is why it is by repressor mechanism. First mechanism is repressor mechanism, and second mechanism is attenuation of transcription. Okay, so in this attenuation mechanism, what happens? If the transcription begins, the rate of the transcription is fine tuned by a second regulatory process called transcription attenuation. So you have to remember this particular sentence. Okay, so here one particular term is there, that is the rate of the transcription is fine tuned by a second regulatory process. So this first regulatory process that we have already discussed, that will regulate the tryptophan synthesis, of course, but the second mechanism once again check the need of the tryptophan synthesis, and it will regulate finely. Fine regulation takes place. Okay, so that is why what happens sometimes, the tryptophan levels are little bit lower than the, suppose if the tryptophan levels are little bit lower than the required quantity, okay, required amount, so then what happens, that genes, structural genes will be transcribed. Okay, so that transcription, because of the transcription, what happens, the translation takes place, and enzymes will be produced, and that will enrich the pool of amino acid. Okay, that leads to abundant amount of tryptophan. So that is why the difference, only slight decrease in tryptophan levels should not lead to the overproduction of tryptophan. So that is why the second mechanism again checks whether really necessary, that tryptophan production is really necessary or not. If it is really necessary, then it allows. If it is not necessary, really not necessary, and there is only a small change, a small decrease, then what happens? It prematurely terminates the transcription process. So transcription process begins, but it will not end properly. It will be prematurely terminated. So that process is called transcription attenuation. Okay, it is finely tuned by the second process. Okay, so for the expression of tryptophan operon, we have two levels of regulations. First level is repressor mechanism regulation. Second mechanism is attenuation of transcription. Okay, so here you can see the in this attenuation process, transcription is initiated normally, but is abruptly halted before the operon genes are transcribed. So operon genes are not transcribed and it is terminated, okay, by the attenuation process. So the frequency with which transcription is attenuated is regulated by the availability of tryptophan and relies on the very close coupling of transcription and translation in bacteria. So to understand the transcription attenuation process, you must understand one more important concept, that is coupling of transcription and translation. If you don't understand what is coupling of transcription and translation, which takes place in prokaryotes only, okay, so you cannot understand the basic mechanism of transcription attenuation. So let us see what is coupling of transcription and translation. Okay, so in case of prokaryotes, there is no specific nucleus because they are prokaryotes. They named like that because there is no separate cell organelles like nucleus, mitochondria, lysosomes, all those things. Okay, that genetic material is present in the prokaryotic cell, and there is no nuclear envelope which surrounding the genetic material or genome or DNA. Okay, so there is no such kind of thing like nuclear envelope or nuclear membrane around the DNA molecule. So the DNA is simply exposed to the cytosol. So therefore here the transcription takes place on the DNA. As the transcription takes place, that mRNA emerges out from the RNA polymerase. By the time the RNA polymerase emerges slowly from the RNA polymerase, that translation also starts. That ribosomes comes and binds to that mRNA, which is being synthesized, not completely synthesized. It is just started. The synthesis of mRNA is started. Just it proceeded to some extent. So by that time that RNA, that mRNA comes out from the slowly emerges out from the RNA polymerase. So now that mRNA is exposed to the cytosolic contents. Therefore, the that ribosomes, tRNA molecules, initiation factors, elongation factors, all this comes into play, and the entire translation machinery binds the mRNA and start the translation process. So now what happens? As the RNA polymerase moves forward, mRNA comes out, and the ribosome and the translation machinery that will move on the mRNA. So that therefore the transcription and translation, both the processes takes place simultaneously. RNA polymerase moves forward, and that ribosomes simply they will follow the movement of RNA polymerase. Okay, but the RNA polymerase is on DNA, ribosomes are on the mRNA that is being synthesized. Okay, so that is what coupling. The transcription and the translation both are being coupled. Okay, so that is what the coupling of transcription and translation. So here you can understand this, like this is the DNA. Sorry, let me draw this. Suppose this is DNA. I will write only single strand for your convenience. And what happens here, the RNA polymerase binds here. This is suppose the promoter region, and it starts the transcription process. So as it moves along this, so from this point it moved to this point. So then what happens, that mRNA that is being synthesized, that emerges out through the RNA polymerase. So now this initial sequence of mRNA contains the Shane-Dalgarno sequence, AUG, and so on. So then what happens now? So now we have RNA polymerase and mRNA that is just emerged out through the RNA polymerase, and this binds the ribosomes. So now what happens? These ribosomes start synthesizing protein. Okay, so that is what coupling. So again I will draw this. This is the RNA. So it started from here. The RNA polymerase started from here and it reached here. So you will get this mRNA, suppose. Okay, now what happens? The ribosome binds like this, and it binds tRNAs and so and so. It moves along like this. Okay, so it will move. So now it reached up to this extent. So then what happens here? The protein polypeptide chain comes out. So now what is happening here? Transcription is taking place, mRNA is being synthesized, and that mRNA is bound by ribosomes, and translation is also taking place. Okay, so that transcription is not at all completed, but the protein synthesis already begun. Okay, so that is why these two processes are being coupled. So now transcription and translation, both the things are taking place simultaneously. So this is what coupling. So now the ribosome is closely following the RNA polymerase. Okay, closely following the RNA polymerase. This is what the coupling of transcription and translation. So this is the basic concept which we have to understand, we have to learn before we understand the transcription attenuation process. So this is the case that that we can see in prokaryotes only, not in eukaryotes. In eukaryotes there is no coupling, because eukaryotes are having a this nuclear membrane around the DNA. So transcription takes place inside the nucleus. After the completion of the transcription only, that mRNA comes into the cytosol, then it undergoes the translation process. So that means transcription takes place inside the nucleus and translation takes place inside the cytosol or outside the nucleus. So therefore those two processes are separated from each other. They are uncoupled. Okay, so now when we look at the attenuation process, you have to understand one important thing, that is the coding sequence. The coding sequence of tryptophan operon, that is the structural genes. Before the beginning, the starting point of the structural genes, we have some preceded coding sequence. That coding sequence is called a leader sequence. It is a leader sequence. So you can also see this leader sequence in the tryptophan operon regulation. Okay, so I will show you here. So this, look at here. So you have these structural genes. Okay, preceding these structural genes, you have a specific sequence called leader sequence. Okay, leader sequence. So this leader sequence also undergoes translation process and produce a length of polypeptide chain. So the synthesis of polypeptide chain starts from here. Okay, first of all, in the polypeptide chain you will get leader sequence, then only you will get the tryptophan E. Okay, so tryptophan E. So that means the first portion of mRNA corresponds to leader sequence. So the ribosomes binds to the initiator site, initiation codon, and they first of all synthesize the leader sequence. Then only they enters the structural gene. So leader sequence is the first sequence that is going to be synthesized from the mRNA. So that is why it is called leader. It is in the first place. Okay, so this leader sequence, when we look at the structure of the leader sequence here, so this is the leader sequence. This entire length of is the leader sequence of tryptophan L, means the leader sequence of tryptophan mRNA. So now in tryptophan mRNA, when the leader sequence in the DNA is transcribed, you will get tryptophan leader sequence in mRNA. So this sequence contains 4 sequences. This leader region contains 4 sequences. Sequence 1, 2, 3 and 4. So one of the important point you have to remember is the first sequence is very important. In the first sequence you have the first sequence codes some number of amino acids. So these are the amino acids coded by the first sequence. Okay, so in this first sequence you have 2 W's. W stands for tryptophan. So single letter abbreviation of tryptophan is W. Okay, so you have 2 codons that will code tryptophan. Okay, you have 2 codons in the sequence 1 of leader sequence, leader region, that codes for 2 tryptophans. So now what happens during the translation process, that ribosome binds to the sequence 1. So it starts the synthesis of protein from the leader sequence. So first of all it enters the sequence 1 and it will read all the codons in the sequence 1. So and one important thing we should note is, among all these codons of sequence 1, 2 codons specify tryptophan itself. So that is why what happens here, if the tryptophan levels are high, then the corresponding tRNA, charged tRNA levels will also be high. So then what happened, the ribosome read these 2 codons very quickly, because the tryptophan tRNA, the tryptophan acylated tRNA, charged tRNA's availability is more. So that is why it quickly read these 2 codons and enters the region 2. Okay, region 2, it enters the region 2. So that is what here. So the importance of first sequence is it contains 2 codons that specifies tryptophan, and the importance of second sequence. Okay, so the importance of sequence 3, in fact sequence 3 is complementary to both the sequences, that are both the second sequence and sequence 4. 3 is complementary 2 and 4. The role of first one is it has 2 codons for tryptophan. Role of third sequence is either it can interact with sequence 2 or sequence 4, because it is complementary to these 2 regions. Okay, so now if the 3 interacts with the 4, then what happens? The 3 plus 4 gives a attenuator structure. If the 2 plus the 3 instead of 4 it interacts with the 2, so then no attenuation takes place. Okay, so this you have to remember in this case. Okay, so now if you look at one situation in which tryptophan levels are high. So what happens if the tryptophan levels are high? That means tryptophan tRNA is also high. The availability of tryptophan tRNA levels are also high. So then what happens here? Here this first sequence, the ribosomes, because of coupling, the ribosomes read this first sequence, sequence 1. Because of high level of tryptophan tRNAs, it read these tryptophan codons very quickly and enters into the second sequence. So now ribosomes have occupied both the sequence 1 and 2. Okay, it is completely occupied by the ribosomes. So now what happens? By this time the RNA polymerase synthesizes 3 and 4. So now the 3 has no other option. If the 2 is free, then it interacts with the 2, but 2 is not free, it is already occupied by the ribosome. So therefore the 3 interacts with the 4 is free now, 3 is free now. So there is complementary sequence between these two. Therefore 3 and 4 interacts with each other and it forms a attenuator structure. You have already studied this attenuation structure, that is stem and loop structure, in case of row independent transcription termination process. Okay, if you don't know about this terminator stem and loop structure, and you go back to that video in which I explained about the transcription termination process. Okay, in which we have that terminator sequence will generate a hairpin like structure that followed by poly-U sequence. Okay, so this causes termination of transcription. Okay, so that is the case 1. If high tryptophan is there, then ribosomes read first sequence and quickly moves into the second sequence. Therefore the 3 and 4 will be free and they interact with each other and form the attenuator structure that causes the termination of transcription. Okay, and if the tryptophan is low, then what happens? At the first sequence the that ribosome will halt. It will take some time to read these two codons because the tryptophan is low, therefore the tRNA is also low. So it will take some time to find the tryptophan tRNA molecules. So by the time what happens, the 2 is synthesized and 3 is synthesized. Now what happens? 2 interacts with the 3. Okay, so 3 now interacts with the 2, so therefore it won't form a attenuator like structure. Okay, so now this 2 and 3 association doesn't act as a attenuator. So now that RNA polymerase easily moves along the structural genes and transcribe the mRNA, and by that time that ribosome also translate that mRNA and produce the structural gene products. Okay, so this is what the transcription attenuation. So at high levels of tryptophan, the ribosomes read first sequence and enter the second sequence, so that the third and fourth base pairing takes place, that forms a attenuated structure. And at the low level of tryptophan, the ribosome halts at the sequence 1, therefore the sequence 2 and sequence 3 interact with each other and that doesn't form a attenuator structure, and it will not prevent the transcription. So now transcription will proceed. So therefore this will check whether the tryptophan levels are high or low. Okay, this is the second level of regulation of tryptophan production. So that is what the transcription attenuation. Okay, so one last simple topic is there, that is negative regulation, positive regulation, and we have already studied what is negative and what is positive regulation. So negative regulation, the lactose operon itself is the example for negative regulation, in which the repressor, so if repressor is involved, so then it is called the negative regulation. So repressor will stop the transcription process, so that is why it is negative regulation. And catabolite repression is the positive regulation, in which catabolite, that activator is there, that activator will activate the transcription process. So if it will positively regulate and it will negatively regulate. So that is what positive and negative regulation. Okay, so if you have a question in your examination, if you are asked to write about the negative regulation, so you have to write about the lactose operon, that's it. And if you are asked to write about positive regulation, then catabolite repression is the answer for that. Okay, hope you have understood this entire unit of regulation of gene expression, and all the best, and thank you all.